All right, everyone, we'll go ahead and get started. Welcome to another session of our evolution seminar series. Today, we'll be learning about primate nutritional ecology as we're joined by Professor Margaret Bryer from the Department of Anthropology here at UW-Madison. So without further ado, let's get to the fun part. Please join me in welcoming Professor Bryer.
Thank you so much. As you mentioned, today I will be talking about primate nutritional ecology, but also primate nutritional ecology as it intersects with social behavior.
Just going to pop up on the screen.
Oh, whoops. Sorry. Yeah. There we go. So I'll be talking about the interaction between social behavior and nutritional ecology. So to give you a roadmap for my talk today, first, I want to introduce you to primate socioecological models. So there's a long tradition in primatology coming up with these verbal models to explain the evolution of primate social behavior. Then I want to talk about integrating nutritional ecology into these primate socioecological models. Then we'll talk about guanins. Hopefully I will get people excited and introduced to this radiation of primates that you may or may not have heard about before. But we'll talk about guanin female social dynamics and nutritional ecology. And then I'll talk about some of the future directions in social nutrition that I'm interested in my research program. So let's start by talking about primate socioecological models. So primates exhibit a wide variety of social systems. So here you have sort of the classic social primate, the baboon, multiple males, multiple females and their offspring living in a social group. Females have an established dominance rank, males have an established dominance rank. And the variety of social systems and mating systems that we see across the primate order are quite dramatic. So while we have some species like baboons or our closest living relatives, the chimpanzees, who live in these multi-male, multi-female groups, there are different dynamics that can go on in those multi-male, multi-female groups. So for example, with wild chimpanzees, they engage in fission-fusion social dynamics. So they'll fission into groups when they're foraging and then fuse back together in a larger group at other times. And then there are a variety of other organizations that primates can come together with, including single female multi-male organization in some South American monkeys like these marmosets, as well as single male multi-female organizations, the classic example of that being the silverback gorilla and multiple females in his social group. And then there are some instances of primates that are solitary or semi-solitary. So here you have this adorable pato, a nocturnal primate that is primarily solitary. There is actually a great ape, though, the orangutan, that has more solitary or semi-solitary tendencies. So you can see this wide variation in social organization across the primate order. And so for a long time, researchers have been coming up with ways to predict how this variation evolved, or think about how social behavior evolved in such a diverse manner within an order. And so first, I want to sort of present you with the simplified verbal model of primate socioecology. This is sort of a conglomeration of several primate socioecological models to give you a sense of the kinds of variables that people are thinking about. And then I'll talk a little bit about the history of these primate socioecological models. So two major drivers of primate social structure have been argued to be predation risk and food distribution. So how likely a primate is to be eaten by a predator and how food is distributed in the environment. A lot of these classic models will characterize food as being clumped versus dispersed. So the idea being that this would drive aspects of social behavior. And so predation risk is going to influence the distribution of females as well as the distribution of males in the environment. And then also is linked to the question of, do females group together with kin? And then food distribution, because of the different reproductive strategies of male and female mammals, is mostly, according to these models, going to influence the distribution of females. So food distribution will determine the distribution of the females in the environment and the nature of those social relationships among adult females. And then these models have made additional links between, for example, the distribution of females in the environment being linked to this distribution of males, generally being the males go where the females are. And then also the distribution of females in the environment influencing the nature of female social relationships. So this is sort of a conglomeration of some of these verbal models, but I wanted to give you a sense of the history of these models. So the earliest versions of these socioecological models really emphasized the role of food distribution and predation. So you have Crook and Gartland, 1966, as well as the classic Richard Wrangham, 1980 article, and Van Schaik's 1989 article, creating this sort of foundational primate socioecological model that really emphasizes predation risk and food distribution, leading to this variation in primate social structures. And then later socioecological models add a whole bunch of other variables. So the students in this class read the Stirk article for today, hopefully. So Stirk added infanticide risk, so the risk that an adult male will come into the social group and kill infants to improve his access to females and improve his reproductive success was added into these models. And then more recent versions of this model have also included disease transmission and information sharing. And interestingly, Romano in 2020 and others have proposed that there's sort of similar mechanisms here between social information and socially transmitted pathogens, potentially affecting social structure and vice versa. So you can see that with time, this verbal model of primate socioecology is getting pretty busy, right? There are a lot of variables being thrown into these models. And to go back to Stirk's version of this model, I wanted to point out that for some of these versions of the model, there are specific predictions about the types of social dynamics that are going to exist among females in a social group based on these variables. So this is a diagram showing you the basic setup of Stirk's model. She has food distribution and predation risk influencing female gregariousness or female social tendencies. And then she also links that to competition among females and that leading to particular types of female social relationships. And they especially in this paper emphasize that you can think about female agonism or the aggressive or dominance-related behaviors among adult females in a social group as being on several different scales or several different parameters. So they can vary in terms of whether they're egalitarian or despotic. So if you have an egalitarian social organization, it's hard to detect dominance rank among the adult females in the group. If you have a despotic organization or despotic type of agonism in a female social organization, it's going to be very clear who's dominant to whom. They also brought up the fact that you can think about dominance relationships and agonism in terms of individualistic versus nepotistic. Again, this is determining how individual females' dominance is related to one another. And then another parameter is tolerance. So how tolerant females are to one another. And as a result of this, they generate several different social categories, one of which is going to become more relevant later in the talk because researchers have proposed that this is what all guanin species fall under. But I'll get to that in a moment. Now, at this point, because this is a verbal model and because there's so many variables involved, you probably are thinking that this might have some limitations. So there have been many critiques of primate socioecological models over the years. Here are just a few. So one major critique that has been made repeatedly is that it's an example of ecological determinism. The focus of a lot of, especially these earlier models, is that ecology is determining everything. And so many researchers have pointed out that we cannot forget about phylogeny and that these early models do. Additionally, and this critique I'm especially interested in with my work, is the fact that the food component of these models is not very specific. So there is mention of food distribution, food quality, but I think we can get potentially more detailed in what that means. And it's also it's quite difficult to test some of these questions about food when the variables involved are a little bit general. And then researchers have also pointed out that when people try to test parts of these verbal models, there are examples in the primate order that don't seem to pan out based on these predictions. So we're not sure how effective the predictive value or how predictive these models are of actual primate behavior. And then one point that has been made by the people pointing out that we need to consider phylogeny more explicitly in these models is that the original version of these models that was really emphasizing predation and food actually was very helpful because it served as a sort of null model. But these later models where we're adding all these new variables are just sort of throwing in the kitchen sink in a way that it's really hard to test predictions based on the models. So with these critiques in mind, I want to talk a little bit about integrating nutritional ecology and something that has been referred to as social nutrition can help with testing primate socioecological models or aspects of primate socioecological models. So as we all know, diet is fundamental to an individual's success, and primates have incredibly variable diet. Here I have the sort of classic fruit, insect, leaf eating primates, but primates are quite diverse in terms of the foods they're eating and also whether they're specialists or generalists. So here is a bamboo lemur eating bamboo that has enough cyanide in it to kill an adult human. This is a very specialized primate and an unusual example in primates of a specialist. And then we have the baboon, which is a very dramatic generalist. And those of you who are aware of baboons in South Africa know that these guys also expand their diet to include human trash bins. So here, this one is eating some bread. And then there are some primates that also eat meat. So chimpanzees will consume monkeys in their environment. And I am specifically interested in nutritional ecology. So remember that all of these foods that I'm describing are parcels of, or rather they're packaged nutrients or combinations of nutrients. So my area of study is primarily nutritional ecology. So we're looking at how an individual gets the nutrients they need to survive and reproduce. And those nutritional components and non-nutritional components involve several things. So you have macronutrients. So macronutrients are chemical components like carbohydrates and fats that can provide energy. Then you have stuff like protein for building and repairing parts of the body. And then micronutrients are also crucial. So these can include various vitamins and minerals. I should note that vitamins and minerals are far less studied in primate nutritional ecology, although we do know quite a bit about sodium because we know sodium is limiting in tropical environments. And these micronutrients are serving a variety of functions in the body as well. And then we also need to take into account what we would refer to as antifeedants. So these could be components that make foods less digestible or are toxic. So there are a variety of plant secondary compounds in tropical environments that can make foods less tasty or potentially act as a deterrent. And then there also are parts of fiber that are indigestible that are also antifeedants. So just to take a step back and think about the variables that these wild primates are dealing with when they're trying to meet their nutritional needs, an individual needs to decide what food to consume in their environment. This is already somewhat limited by their gastrointestinal tract. So whether it's the structure of their GI tract or the microbes that are in their GI tract that can help or hinder their ability to digest certain foods. And the nutrients available in the environment are determined by a variety of abiotic factors, including soil composition, water, sunlight, climate. And so when they're going through their environment, they're choosing between a variety of foods that are packages of these nutrients. So if they're choosing ripe fruit, those tend to be higher in sugars and fats, more readily available energy. Leaves tend to be higher in protein and have some digestible fiber, which can be used for energy. But because part of the plant cell wall is indigestible, there's also an antifeedant component to these leaves as well in the indigestible fiber. And then insects are also a big part of some primates' diets. And depending on the developmental stage of an insect, you can get very different nutrition. So if these monkeys are finding larvae of particular insects, then those are going to be quite high in fats. If they're finding adult insects that they're consuming, high in protein, but also high in an indigestible compound called chitin. And then what I want to add to this discussion and link back to the socioecological models is that there's also a social component here. If there are other individuals in your social group present when you're trying to find these nutrients, that's going to potentially affect your nutritional strategy. So whether that's conspecifics or members of other species. And so this leads to this elaboration of social foraging theory that researchers have proposed as social nutrition. So this is an idea put forward by, or a framework put forward by Stephen Simpson and David Robenheimer at the University of Sydney, and then Mathieu Leheureux at the University of Toulouse. And this is partially based off of Simpson and Robenheimer's geometric framework way of thinking about nutrition, which is basically a framework in which you can consider multiple nutrients at once. And then Leheureux, in collaboration with them, has proposed this social nutrition model, primarily in the context of insects, especially eusocial insects. And so the idea here is that there's this intuitive point that feeding shapes sociality. So where the food is, their individuals are going to congregate, and that's going to affect social dynamics. And then sociality can also influence feeding and nutrition. This potentially can be in the context of dominance hierarchies. If more dominant individuals are better able to access higher quality food, then that has implications for their fitness. And then in a mixed species context or a polyspecific association context, this gets even more interesting because you have social dynamics within species, but also social dynamics between species that might influence nutritional strategies. And as I mentioned, this framework of social nutrition has primarily been tested in insects, and especially in the context of bees, which is the set of species that Leheureux works with. And there have been some simulation models that he's run to make predictions about social nutrition in vertebrates, but they haven't been explicitly tested. And so launching off some of these simulation models that Leheureux put forward, I want to sort of present how this framework would predict that how social nutrition in a competitive context might work with primates. So the way this is set up, you have one nutritional component on the x-axis and one nutritional component on the y-axis. So you have protein on the x-axis, carbohydrate on the y-axis. And you can think of this as a series of meals that an individual is eating. So here you have your individual, and with each meal, they are moving towards an intake target. So each of these rails here represents a food. Let me just get the laser. Okay, so each of these represents a food. So here you have a higher protein, lower carbohydrate food of leaves. And on this rail is representing a ripe fruit, so higher carbohydrate, lower protein food. And so the animal is mixing these two foods through a series of meals to reach this intake target. And the intake target is this hypothetical target that the animal needs for these nutrients to be able to survive and reproduce. And so you can imagine that in a context with conspecifics or other members of your group, or in a context where you're in mixed species association, this might be altered. And if we think about this on a daily basis, which is how I'm going to consider it past this slide, on a daily basis, this reaching the intake target might become more challenging. And generally, based on some of these simulations that Leheureux has done, the argument is that with conspecific or heterospecific competition, there might be just an overall decrease in intake of both of the nutrients, or there might be a sort of spreading out of what the individual is able to eat in the nutrient space. And this effectively means, for example, in the context of competition, this individual is going to start feeding over here because they're only able to eat leaves because they can't access the ripe fruit. Okay, so you can sort of think of this as like a nutritional space that the animal is moving through, and there are different variables that might affect their nutritional strategy. All right. So the primates with which I want to test some of these predictions belong to a group of primates that I want to introduce you to that are pretty exciting radiation of primates for evolutionary biologists. I think there are many evolutionary biologists who are interested in them for a variety of reasons. I'm especially interested in their social dynamics. But guenons are one of the most diverse clades of living primates, and they radiated about 10 to 15 million years ago, and today they include over 30 distinct species. And so you might notice from this image by my colleague Will Allen that there's a lot of diversity in facial features, in skin pigmentation, pelage color. So that's sort of the first noticeable thing about these animals is that they've evolved a wide variety of static visual signals in their face. These monkeys tend to draw attention to these static signals with not only coloration in their face, but also in their genitalia and through various movements like head bobbing or presentation of their behinds. And I thought these illustrations by wildlife artist Jonathan Kingdon are really good at showing this sort of dynamism in how movement and color play a role and how variable they are. And speciation in guenons has often involved alterations, as I mentioned, in skin pigment, also in vocal phrasing. So there are actually closely related guenon species that have evolved distinct vocal patterns. And so despite these pretty dramatic physical differences, they're actually quite difficult to distinguish based on skeletal features. Also, in some cases, genetically, they're more difficult to differentiate. And so here is a taxonomic overview of guenons, with most of the species falling into the Cercopithecus genus. And there you can see that there are also other genera in the guenons, some of which are more terrestrial. The Cercopithecus genus is primarily arboreal. And this map is showing you a very wide distribution of these animals. So this is a very successful radiation of primates in sub-Saharan Africa. And they often live sympatrically. So this is actually showing you in darker colors where there are more species of guenons overlapping. And then here are some phylogenies of or taxonomies of guenons. So on the left is a species tree based on autosomal gene trees, and on the right is a tree constructed from mitochondrial genomes. You can see that there's some discordance between the two trees, but the main takeaway here is that there are multiple clades that are terrestrial and multiple that are arboreal, and the Cercopithecus clade is primarily in this arboreal section. And for the results I'm going to show now, I'm focused on a particular species of guenon that falls in the Cephus group. So it's in the arboreal subgroup of guenons. And they are called the red-tailed monkey. So for this particular species, I went to Kanyawara, which is a site in Kibale National Park. This is in southwest Uganda. It is a tropical mountain forest, and it's run by Makerere University, has been a long-term field site, so 45 years. And there are 13 species of primates there, including 3 guenons. So we have red-tailed monkeys, blue monkeys, and loise monkeys. And so what I'm going to talk about now is testing social nutrition questions with these red-tailed monkeys. And so these guys are small-bodied arboreal omnivores, so they switch between a variety of foods. This figure is showing you a bunch of different food parts that they eat, broken down by daily percentage in kilocalories in the black bars, and percentage by feeding time in the white bars. So things like fruit, young leaves, exudate or gum, and insects are really dominating their diet, but you can also see that there are a variety of other foods that they're exploiting. And the diverse diet is arguably facilitated by, one, their cheek pouches. So these are cercopithecine primates. So like other cercopithecines like baboons and macaques, they have these cheek pouches where they can store food temporarily and then process it later, and they can do some initial digestion with enzymes in these cheek pouches. Joanna Lambert found that they also have a longer retention time than you would expect for an animal of their size. So based on experiments looking at how much time the food is actually in the gut, their food is in their gut for about 24 hours, which is longer than you would expect for an animal of their size, and suggests that there's potentially some sort of hindgut microbial digestion happening. And red-tailed monkey social groups include multiple philopatric adult females. So females stay in their natal group, and then one or multiple adult males. And the guanin within group social structure is not very well understood in red-tails or actually across these species that I've been talking about. And one thing we do know is that there's a lot of between group aggression. So adult females are especially active in fighting other groups over high value food sites. But the relationships among females within groups is a little bit more ambiguous. And Stirk, their socioecological model, proposed that all guanins are, quote, resident egalitarian. So resident because the females stay in their natal group, and egalitarian because it was hard to detect any sort of dominance relationships between females. But Stirk's paper is theoretical, so that was not tested. And so there is some ambiguity there. Are all guanins resident egalitarian? And there's some research with blue monkeys, another Cercopithecus guanin, that suggests that, not so fast. Maybe there are just more difficult to detect dominance hierarchies happening among female guanins. So Class and Cords and Marina Cords more generally has had this long-term blue monkey project in Kenya and has actually detected moderately despotic, tolerant, and nepotistic dominance hierarchies in adult female blue monkeys. So this suggests that perhaps there's more there. And so I studied 3 habituated red-tailed monkey groups at Kanyawara. So shown here are actually feeding trees at Kanyawara marked for the 3 different group home ranges. And the boundary of the park is indicated here. And so this group indicated in red actually ranges at the edge of the park, and the star here is the biological field station. So unlike these 2 groups, the group in red ranges in an area that overlaps with the field site and also overlaps with an area that's recovering from clear felling of plantations. And so this is a slightly different type of habitat than the other 2, which are in primary and secondary forest. And so I hypothesized that individual rates of female agonism, so when I say agonism, again, I mean not only chases, attacks, but also things like when one individual female supplants another one, takes her spot, or displaces her. So individual rates of female agonism will drive daily macronutrient energy intake. And then I also hypothesized that group membership will drive daily nutrient intake as well. So this has to do with the slightly different habitats that the 3 groups are ranging in, specifically that one group that seems to be in a much more disturbed area. Oh, I'm sorry, that didn't pop up. Yeah. And then I also made a prediction about mixed species association. So I mentioned that mixed species association can complicate this social nutrition picture, right? So at Kanyawara, previous studies have actually shown changes in feeding behavior when multiple species of monkeys are feeding in proximity. So I mentioned that there are other guanins. So there are blue monkeys there. And then there also is a non-guanin, a mangabe, a gray-cheeked mangabe that's there, but has a pretty overlapping diet. And so previous studies have shown that there seems to actually be spacing based on body size in where these species forage in the trees, which might have implications for an interspecific dominance hierarchy that might affect who gets the highest quality food. And so I hypothesized that association during feeding so red-tailed monkeys associating during feeding with blue monkeys and gray-cheeked mangabes would affect their daily macronutrient and energy intake. And in terms of field methods, so with this study, we did full day focal follows of individual adult female red-tailed monkeys. So with a team of 3 to 4, we were monitoring what that individual was eating all day, as well as her social interactions with other adult females, as well as whether or not the group she was in mixed species association with mangabes or blue monkeys. And in addition to collecting behavioral data on the individual red-tailed females, we also were tagging trees and shrubs from which she was feeding, and collecting samples at the same level of the tree or shrub, same tree or shrub, and then going through a field protocol, which I can go through quite quickly. I can talk more to the students about the details of the methods during the class session if you'd like. But we measure wet weight, unit weight. We dry them in a dehydrator, which is the same kind of dehydrator you guys would use for, drying jerky. Then you measure a dry weight, grind it up, and then export it back to the lab. And I did just want to emphasize the variety of foods that we were doing this with. So these are all examples of red-tailed monkey foods. So everything from flowers to gum to leaf galls to various fruits, and then insects, including cicadas and noctuid moth larvae. So it's a whole variety of foods that we are preparing for nutritional analyses. And then we ran nutritional chemistry assays at the Wildlife Nutrition Project lab at Hunter College. So basic macronutrient analyses, as well as mineral analyses. And we also use near-infrared reflectance spectroscopy, or an NIRS, for running leaf samples specifically. So this is a technique that's long been used in assessing agricultural feed quality, and so it's been incorporated into primate nutrition work as well. Although having really nutritionally variable foods makes it harder to use a machine that requires you to calibrate it for, quote unquote, fruit. So results. So female red-tail agonism did not affect daily nutritional intake. So we looked at agonism received and agonism given and did not find an effect. I should take a step back and say that there was not enough agonism to try to construct dominance hierarchies. That said, this was a short-term project. Part of my longer-term social nutrition project is getting longer-term agonism interaction data to be better able to detect dominance hierarchies. But for the purposes of this phase of the project, we were not able to find an effect on daily nutrient intake based on agonism. And just to give you an idea of how little fighting happens with red-tailed monkeys. So reminder, we're talking about not only contact aggression, non-contact aggression, but also things like displacements. So in primates generally, you have a lot of variation in how much fighting goes on between adult females. So at the really high end, you have female chacma baboons, where they're basically engaged in some sort of agonism, whether it's displacement or some form of aggression, every 20 minutes. And then at the other end of the spectrum, you have certain howler monkeys where adult females might only engage in these agonistic interactions every 100 hours. And red-tailed females fall into one interaction every 7 to 8 hours. So not quite as extreme as the howler monkeys, but still not very much fighting. I was in the field with some chimp researchers who got to see lots of fighting every day. And I came back from the field one day and said, a red-tailed female pushed another female from the branch. And they all looked at me like I was crazy. But it was a really big deal because they don't do contact aggression very much. So why might I have found this, that daily intake in macronutrients and energy wasn't affected? So one possibility is that competition among adult females is buffered by things like group spread. There's a variation in how spread the group is during the course of the day. Additionally, previous researchers have pointed out that red tails and other guenons switch between foods really rapidly. So maybe that is also buffering the competition. If there's always an alternate food source that you can switch to, it could reduce the amount of potential competition. And then there's the fact that perhaps feeding competition between groups is more relevant to individual nutritional shortfalls or advantages. So there's the fact that red tail females are definitely aggressive between groups and are defending high quality food patches. So maybe these within group dynamics are just not as important to these nutritional shortfalls. In terms of habitat variation, so I mentioned that there was slight habitat variation between those three groups. And so I looked at the nutritional strategies, the average female nutritional strategy for each of these groups, and found that one of them is a little bit different. So this is a right angle mixture triangle, which is another nutritional geometry way of showing multiple nutrients and how they're balanced. So you have protein on the x-axis, non-structural carbohydrates and fat on the y-axis, and then digestible fiber on the implicit axis. And the average intake is represented so the average intake per group is represented by these three diamonds. And you can see that the yellow diamond is a little bit separated. This is the group that's in the more disturbed habitat. This is the one that was ranging in the biological field station and in the recovering from clear felling area. So you can see that there's more carbohydrates and fats intake demonstrated here compared to the other two groups, and slightly less protein. So what's going on here? When we looked more specifically, it became clear that one food source actually, or one species actually, had a really big effect on the nutritional strategy of that one group. So Prunus africana is an endangered tree species that has some features that make it very high quality food, and also it is found in disturbed habitat more frequently. So Prunus africana undergoes gummosis almost constantly. So there's tons of gum coming out of, or exudate coming out of this tree. Nutritional analyses showed that this gum is really high in complex carbohydrates, and the red tails and other animals in that area really enjoy that gum. The fruits of this tree are especially high sugar compared to other fruiting trees in the area. So that also can contribute to the increased carbohydrate intake of this group. And so the presence of this tree in that more disturbed area is clearly influencing the nutritional strategy of the individuals in that one group. And then I did just want to make one point about minerals as well. So though all three groups of red tails were reliant on insects for sodium, so this figure is showing you food items, percentage of the daily sodium intake, and then percentage of grams sodium intake in white shown here compared to percentage of feeding time. So they're getting a lot of sodium from insects, which in itself is interesting, but there also is a lot of sodium from exudate or gum. And that is really driven by that group that has a slightly different nutritional strategy and is really reliant on Prunus africana gum. The Prunus africana gum was a major sodium source. And as I mentioned earlier, sodium is limiting in tropical environments. So this is something where there seems to be a slightly different mineral strategy for this particular group in a disturbed habitat. So we found habitat variation even at one site can lead to flexible nutritional strategy within a species for this guenon. And then for my, the hypothesis about mixed species association, association with blue monkeys or gray-cheeked mangabeys did not affect intake in red-tail monkey daily macronutrient or energy intake. Even if there was fighting, even if there was chasing from blue monkeys, it did not seem to affect daily intake of red-tail females. And so again, the question is, why wouldn't this influence nutritional intake? So again, it might be those kind of competitive buffers that I was talking about in a conspecific context. So group spread would reduce competition. Food switching could reduce competition in a heterospecific context as well as a conspecific one. And then I mentioned the fact that there seems to be some sort of pre-existing dominance hierarchy among the monkey species at that site, where larger bodied individuals tend to be feeding higher. There seems to be some stratification going on or some sort of established interspecific hierarchy that might be dictating the kinds of foods that the red tails have access to. And so maybe that already determines this. And so there isn't this effect that we would see. So in summary, when I'm looking at these red tail monkey social dynamics and nutritional ecology findings, this is a rare examination of social nutrition in a vertebrate. So I'd mentioned that this social nutrition framework has primarily been tested in an insect model. And there have been theoretical predictions about what would go on with vertebrates, but I think there's really rich avenue for looking at or testing socioecological questions with this social nutrition framework. We've gained more information about the social structure of a guanin, a successful group that we know less about socially. And then the mixed species association finding can contribute to our understanding of nutritional niche separation in a rainforest community of primates. And so the fact that I did not find an effect on daily nutritional intake in mixed species association contexts might indicate that there's sort of a ghost of competition past situation going on where something has already been established. And so we don't see these present day costs. And in the context of evolutionary anthropology, there's also the argument that we can use these kinds of social nutrition studies in conspecific and heterospecific contexts to aid in reconstructing fossil primate feeding strategies. And so now I want to talk briefly about the future directions in guanin social nutrition that I'm excited about. So as you noticed in this beautiful combination of faces of guanins, there are a lot of guanins out there. And so in addition to red-tailed monkeys, I also have started a project on loise monkeys. And to orient you to where these guys are in the taxonomy. So unlike the red-tailed monkeys, which are in that sort of dominant Cercopithecus arboreal group, the loise monkeys are really interesting because they are terrestrial or semi-terrestrial, and have recently been pushed into this other genus. And so I think that introduces an interesting comparative case where you can look at not only gaining more information about this intra-group social structure of these two guanins, but also can compare a terrestrial and an arboreal guanin in terms of these nutritional and social questions. And these two guanins occur sympatrically at two sites in Uganda where I'm working. So in addition to Kanyawara site, where I was just showing you the red-tailed monkey results, there's also a site called Bwindi National Park that is also in Southwest Uganda, but is has a slightly different plant composition and environment. And so we're doing a comparative study looking at red-tailed monkey social nutrition at Kanyawara and at Bwindi. And I did just want to do a plug for my lab because I started my lab in 2023. I am an assistant professor in the anthropology department. And I am looking for graduate students. So we do a combination of fieldwork in Uganda and South Africa. I didn't refer to the South Africa project today, but primatology fieldwork abroad. And then we also have a nutritional chemistry lab here at UW. So the idea is to combine lab and field methods in looking at social nutrition in guanins and other primates. Thank you all so much. Yes.
So you mentioned that food switching might be a way to dampen the effects of agonism in females. Should we therefore expect species that have a more specialist dietary range that effect would be stronger because they have less of an ability to switch to another food?
Yes, absolutely. Yeah, I would think that, yeah, I think that the flexibility of guanin diet, I mean, at least red tails and other guanins where it's been shown that they switch a lot, I think that has a lot of implications for the ability to adapt to changes in the environment.
Is there a guanin species that is a little bit more specialist that we could test it?
So the guanins that I'm aware of are pretty generalist. So yeah, that's an interesting question though, if across those species, from what we know in the published data and what I've looked at, whether we would find one that would be more specialist, I'd have to think about that a little bit more. Yeah.
Jody, talk.
Thanks.
I don't often think too much about like evolution of behavior or sociality and stuff like that. So do you see a lot of variability in social structures when you look across groups within the same species? And does it say something about like plasticity of these social behaviors? And I think broadly, like, does it say anything about evolution of social structures
Yeah.
over biological?
Yeah, so actually, my colleague in anthropology, Karen Stryer, has written a lot about this in primates, thinking about plasticity and social behavior across primates. And there are some really interesting studies looking at just interpopulation variation and the types of flexible behaviors that happen. One study that is not a Gwenin study that I was involved in sort of speaks to this a little bit. So I was involved in a study looking at evolution of social behavior in chimpanzees. And so there are sex differences in chimpanzee social behavior that are very dramatic. So adult male chimpanzees are highly social with each other, and then adult female chimpanzees are, in the wild at least, are thought of as not very social, not very bonded. And so in collaboration with Alex Rosati at Michigan, we were looking at sanctuary chimpanzees where food resources are readily available in a sanctuary context. Reproduction is also reduced because the individuals are on birth control. And so there's this argument, if females are getting food consistently and are not dealing with reproductive stressors as much, would their social behavior change? And in that study, we found that adult females were grooming as much as adult males in the sanctuary context. So that was quite flexible. Aggression remained very similar to the wild, implying that that is not as flexible. And that might be due to endocrine drivers and other, biological drivers there. And then we actually found that some of the adult females in that group were higher ranking than some of the adult males, which never happens in the wild. So there definitely were dominance dynamics that were more flexible. So I think there are really interesting studies in primates that look across different wild contexts, but I think there are some interesting sort of natural experiments we can do in captive contexts compared to wild contexts as well to look at that, the extent of that flexibility. Yes.
I have two questions. One might have part A and part B. About the red-tailed monkeys. There's those three groups. So my first question, I hope is somewhat simplest. How do you define those groups? And then the second one is, if you look at the data points right on the borders of the groups, is there anything that would stick out differently about conspecific interactions on those borders? If you, for example, group the data by observations on fruit trees at the borders. My second question, if you want me to go into it, or if I want me to wait.
Sorry, just could you clarify? So this is for this one? No, which sorry, could you clarify the first question?
Oh yeah, the first question just, I noticed you have those three groups in your study area.
Mhm.
And my question was just, if you looked at data points right on the board, like what makes those groups?
Oh,
And then if you group data by fruiting trees right on the borders, for example, is there more conspecific competition on those borders, and how might that affect this phase? That's sort of my first.
okay. Yeah, so that is a very good point. So these are data points, are tree data points. And so there are instances where the groups will both be interacting with a particular tree. So this is a little bit artificial in terms of how the groups are very distinct in where they're ranging, and there is a lot of intergroup conflict when they interact. But there are definitely instances where one of these trees might be used by multiple groups. And I think that that's a really good idea, thinking about potentially in the future, looking at those trees along the border and how much conflict might arise in those areas, but I haven't looked at that yet. Yeah.
Starting to get a little bit I work in vice, so very different animal, but I'm trying to start thinking about the intake of nutrients, and it's really helpful. Thank you. And one question I have is when I think about what a mouse eats, I think it might be driven, like you said, like anthropologically by drive, like something it needs, you know, sodium versus protein versus
Mhm.
fat. When I think about primates, I think sometimes I eat something because it tastes good, right? Like candy or whatever, and I guess maybe it's YouTube videos. I have this belief that monkeys might be similar to me. They're always driven just by maybe this biological sense of, you know, lack of fat. And so I guess what I'm wondering is in your nutritional part of your lab, where you look at the composition of the material taken in by these primates, is there any comparison to what comes out, like in their fecal boli? Like, are they, you know, if you identify a certain amount of protein and fats in leaves and insects, are they actually absorbing that, or could they possibly be eating it because of taste or color, not necessarily absorbing that? Any thoughts
Oh,
about it?
yeah, that's a great question. So in terms of absorption, so right now the only nutrient where I'm looking at absorption is digestibility of fiber. So we have a series of assays where we're doing sequential fiber analysis for the food and then sequential fiber analysis for the feces. And then that enables us to have a coefficient that we can incorporate into how much of the fiber is actually digestible fiber. But your question about, you know, why individuals choose foods is really interesting because, I mean, there's a whole area of research looking at sensory ecology and nutrition in primates. And I think that there's a lot there. I mean, there are definitely instances where primates are drawn to certain foods because of, you know, certain taste features. And I think that that becomes super obvious in anthropogenic environments where you have, you know, baboons seeking out, I mean, so in my South Africa site, like baboons seeking out empty chip bags because of the salt inside, or, you know, or something really delicious that somebody threw out. Taste definitely plays a role in part of it. So, but yeah, sort of differentiating that seems a bit challenging. Yeah. Yeah. But do you think that your mice never seek something out because it's tasty?
One, obviously work with a very lab around the wild. I pretend that I can say something about their ecology, but they do come from an island where the ecologist colleagues have shown that they spend 4 months in the year feeding on nesting seabirds.
Oh,
And so that's very different fat, carbohydrate profile.
yeah.
But so to answer, maybe those are tastier, or they are. It's almost like us getting a fatty meal. I don't know. At the same time, there's evidence that might boost their density, like the population density here, so it could be good by need. But your question to me is fair as well. It's clearly on my mind.
Yeah.
Yeah.
Thank you. Yes.
I don't remember if you mentioned when the data was taken, but I'm curious if you know anything about seasonality in terms of nutritional ecology and how it affects what is available and what are likely to pick, and also even if there's things like feeding seasons and being with children or anything that affects.
Yeah, great question. So I didn't mention this, but we do a whole bunch of food availability data collection for these nutritional studies as well. So we have a tree transects that we're monitoring for food availability, ripe fruit, unripe fruit, young leaves, mature leaves. And then making sure that as we're analyzing the nutritional intake, we're taking into account food availability as well. And there are fluctuations in how, you know, fruit is available in certain peaks at Kibale. And so we do see a change in nutritional intake by season. We also made an effort to look at insect abundance availability. I don't know how you look at insect availability for a primate in a tropical environment. If you have any ideas, please talk to me, because I feel like it's extremely challenging to quantify. I collaborated with an entomologist at University of Turku, Topani Hopkins, and he was looking at just parasitoid wasps and found dozens of new species of parasitoid wasps. And so the idea of trying to look at the insects that are available to arboreal monkeys in a way that's systematic seems impossible, but I'm sure it's not. I'm sure we can all come up with a solution. But was also trying to track insect abundance as some sort of measure of what insects would be available. So we had these variables also in the analyses of the nutritional intake. And then in terms of mating season, there isn't really for guenons a mating season. There are some birthing peaks that have been pointed out previously. But I mean, these groups are somewhere, they range between about 18 and like 40 individuals before there have been some fissions. And generally there are offspring around year round. Like there doesn't seem to be a very dramatic mating or birthing season system.
So for the things you do have abundance information on, do you try and normalize intake to the amount that is available at each time, or have you taken it into
So
we're eating more fruit, but that's they could just put more fruits around, or
well, so we added, so fruit, for example, we had a fruit availability index or a young leaf availability index. We were incorporating that into our models so that we could make sure we weren't just detecting that effect for example, carbohydrate intake.
That was a very basic question, not a primatologist. You mentioned within group and between group interactions. I'm just curious, to what extent do within group dynamics influence how groups interact with each other? Like if you have one group that's despotic and one that's egalitarian, does that influence how they interact with each other? Or is there just a blanket out group response? Is that species specific? Is that just not known?
Interesting. Yeah. So, I mean, well, so in these early socioecological models, there is this assumption that when you're dealing with, you know, two guenon groups, you're going to have more, you know, egalitarian behavior within the group, partially as a bonding mechanism so you can fight other groups, right? But are you thinking in a conspecific way or multiple species? Because that would be really neat. Yeah. Yeah. But that is an interesting point that, let's say down the road, we have enough agonism data for these three red tail groups to determine that one has more despotic dominance hierarchy. Would that also influence their between group behaviors? I think that would be really interesting. I'm not aware off the top of my head of a study that I can point to for that, but I may be forgetting.
Well, if there's no more questions, let's give Professor Bryer a big round of applause.